Do you hear me? Please tell. Yeah, just drop me a message that you hear me. Wait a second, I'll try to figure out whether I'm online. So great. I have a confirmation that everything is fine and you hear me. So let's go ahead. Well, first of all, it's my second experience in webinar. And my name is Alexander Kotler, as you see here, and I'm working at Tel Aviv University, which is located in a beautiful place here in the Mediterranean. And you see the reflection of this sky. It's a sunset, and you see pink color of the clouds, which are reflected from the wet sand. Well, actually, I want to say a couple of words before I start talking about AFM imaging of DNA structures. I would say that webinar, to my mind, is a great thing. Because, you know, I have been just driving to university today in the morning. Well, a lot of traffic, you burn the gasoline, and you get nervous because of the traffic jam. And it's so pleasant just to sit in your office or in your living room and broadcasting your lecture to the students, which are somewhere. And it's great. I think it's the future of the lecturing in the university is also, something like webinar. So the lecturer just have its cup of coffee, he's relaxed, and he's just giving his lecture, it's a fun. So, okay. So here we're going into the AFM business of DNA-related structures. Why AFM and DNA? What brought me into this field, which I consider a fascinating field? So this actually, I was brought here by accident. My background is biology. I'm coming from, have been graduated from the Department of Biochemistry of Moscow State University a long time ago. And I have been dealing with various things which are mostly related to biochemical reactions and enzymes and whatever. And at a certain stage, I was, again, as I said, I was brought accidentally to more physical studies. I was introduced to Professor Dani Purat from Jerusalem, and he said, why won't we try to measure DNA conductivity? Well, of course you know that DNA is made by Lord not to conduct electrical current, but some calculations say that it might conduct. So we started our collaboration, and before going into details of our studies, and I will present it in 2 minutes, I want to say that I enjoy it very, very much, especially AFM imaging. And I will try in this presentation, I will try to show you the beauty of AFM and how AFM can be applied to biomolecules, because there is a gap between biologists and physicists, and I will try to cover a bit the gap. I would say that what information AFM imaging can give to biologists, especially in DNA field, but not only. So let's go to business. AFM imaging DNA-related structures. Well, again, as a motivation, initial motivation is DNA conductivity. You can see here a DNA wire lying in between two gold electrodes. These are gold electrodes, and this is DNA, and the electrodes are connected. So this wire, DNA one, is lying in this valley, connected to electrodes. And this is, I will not touch upon the conductivity issue, but I will mainly talk about production of those wires and, of course, AFM imaging. And again, what value AFM gives us, biologists. And that's, so go ahead. So there are 3 structures which I'm going to introduce. This is an overview. This is just a very brief sketch of what I'm going to talk about. The first wire is duplex, there's a triplex and quadruplex. The difference is the number of strands. In duplex we have 2 strands, triplex 3, and of course in quadruplex this is a 4-stranded structure. I will elaborate on the details of just a bit later. So let's start from the simplest one. Well, I would say this is the major one. The majority of genetic material in our cells is in this form, which is called B form or Watson-Crick form, because it was initially discovered in 33 by Watson and Crick. It's a double-stranded molecule where two strands are held together by means of hydrogen bonds here in between these pair of nucleic bases. And you can see here there are two possible pairs, G, C, and A and T. And these are the hydrogen bonds in between them. So now I'm going further. Triplex. Well, it's similar to duplex, but the only one difference is the third strand, which you can see here in red. It just merges into the structure of the duplex. Again, what holds it together with the duplex is the number of hydrogen bonds. You can see here the hydrogen bond. The third base, which belongs to this red one, forms with the initial duplex. So this is a triplex. It's much more rigid. It's much more stable. And I want to say that all those structures I'm talking about, they have a relevance to biology. It's not just an artificial structures. They have very, very high importance for the living cell. They're participating in the process of aging, of the cell cycle, of the cell death, of cancer. So these structures are extremely important. It's not only physics. So now this is the last one, not the least one, of course, that I'm going to talk about is a quadruplex. Here you have four strands because red, yellow, blue, and green. These four strands running in parallel to each other, forming this kind of, and the unit here is just a tetrade, meaning there are four bases in the tetrade, and each G, only guanines form this kind of structures, and Gs are interacting with each other, again, by means of hydrogen bonds here. You can see there are 8 hydrogen bonds, 1, 2, 3, 4, 5, 6, 7, and 8 bonds inside this tetrade. And in the middle, you can see the cation, which can be either sodium or potassium, which stabilizes those structures. So these are three structures, main structures I will touch upon during my talk. And those molecules we consider and we want them to conduct current, so we will term them molecular wires. Now, we'll go ahead and say, well, how we make them, how we produce them. So before imaging, you should make those molecules. I mean, we use polymerase. This is enzyme. Of course, I'm an enzymologist. Well, I like very much to use enzymes in my research. So this is an example. Well, the classical mechanism of the polymerase includes extension of the primer, which is a single strand, which is connected to a template, which is, again, a single strand, but it is longer. So what polymerase does is extends the primer just till this end, and placing bases, and the lore says that all the time you find A opposite to T and G opposite to C, as I said, because this is a complementary law. So this is the function of the polymerase. It's just a classical mechanism of polymerase catalysis, which was discovered by Paul Karnberg from Stanford. No, this is not the way we are making our wires. Again, we use polymerase, but the mechanism is different. So we discovered 7 years ago that polymerase, in addition to the catalysis I have been showing you, can do another kind of job. So we just take this, I would term it a template primer system, which consists of, let's say, 10, 12, 13, but it's a small amount of G's in row. This is a double-stranded structure, which is extremely small. I would say here is 10, only 10, or 12 maybe, bases. Oh, this is G and C, only G's in one strand and C's on another strand. So we can order this kind of oligonucleotides from chemical company, and we just prepare this kind of template primer, which is a double-strand, very short molecule. And what we do, we mix this template primer with 2 GTP and CTP, these are nucleotides, with 3 phosphates, and we add polymerase. And what's happened is that polymerase extends this very short initial template primer system into extremely long wires, extremely long molecules. The length of those molecules could be as long as microns. So this is the reaction we have discovered, and of course we used it in our research. So you can see here the progression of the reaction in time. So We start from extremely very, very long template primary system. And you see that in time, the length of the molecules is increasing tremendously. So you can stop reaction anytime. And depending on the time passed since addition of enzyme, till then you stop it. The length can be determined, and so if you stop the reaction 90 minutes after initiation, you can see that the molecules will 3 kilobase pairs long. If you do it after 30 minutes, it will 1 kilobase very long. So you can withdraw the sample in time and depose them on the surface, and you can see that the molecules are there. These are double-stranded molecules which were imaged by AFM, and you see they're rather uniform. Now we can prepare much more uniform structure, and here you see the electrophoresis, the dependence on the length, and here the markers are on the time passed since the initiation of the reaction. And here is the magazine we have published this work. It's Nucleic Acid Research 7 years ago, as I said. Now, we are getting, I will just say where we have some questions, not yet, so I'm going further into the direction of triplex. Now, what's happened? We synthesize this long duplex. One strand is made of G's, another of C's. So, what happened? How we synthesize triplexes? Again, this is the third strand joining this duplex, and it's here, you can see it in red, and duplex you can see in green. So we start with this long, double-stranded molecule, long duplex, and what we do, we mix it with GTP and with polymerase. And what's happened, then polymerase built here a very short extend this G strand, so the overhang here forms. Now, the second phase, it bends here in this direction, and polymerase adding a number of successive cycles of G addition to this strand brings its end right here. So you form here a triplex. Now, the question, it was a proposed mechanism. We want to confirm it. Here comes a stage. I want to show you, to demonstrate in this particular example how AFM can be useful for elucidation of the mechanism of enzymatic reaction. Here you can see the snapshots of the reaction. This is the start. No polymerase added. You can see these are initial double-stranded molecules. You know, they're noticeable, but they're just, they're not very, very bright, I would say. The brightness in the AFM is a reflection of the height or the rigidity, I would say. So now those double-stranded molecules, you can easily see them, but they're not very pronounced because they're not that bright on the image. So we add polymerase, wait for some time, and then withdraw the sample and deposit on the surface, mica surface, and image. You can see the formation of a bright area here on the molecule. So this is because you partly form a triplex. And the triplex is much more rigid compared to double-stranded molecule. That's why you can see it as a bright area. Now, we go ahead. We continue the reaction. And finally, we got there. So we completed synthesis of the triplex. And all molecule, all the way from this end to that end, it's bright, meaning that it's triplex. It's more rigid. So this was a confirmation, the kind of confirmation of the mechanism of the synthesis of the triplex synthesis, which is based on AFM. And it's a direct, evidence for this kind of mechanism. And we made a number of snapshots. You can see in details how this part, bright part, is increasing. It's going like there in time up until it reaches this end, and the whole molecule becomes a triplex. So now we're coming to the last wire, the third wire, which is which is quadruplex. Well, quadruplex means 4 strands. In order to bring 4 strands together, you should have a kind of idea how to do that, because they will never endeavor in solution, will never form this kind of structure. So here I want to present a very smart idea, which is not mine, which was brought by my PhD student, very smart one. The idea is to use Avidin-biotin system to bring 4 strands together. What Avidin is, it's a small protein which is unique in so that it contains an active site, or a site for binding of a small organic molecule cofactor called biotin. This is a biotin structure. It's getting there interacting very tightly with those amino acid residues inside the pockets, and then the interaction is extremely strong. You once bound, it will never can exit this side. I would say almost never. It's not a covalent binding, but it's extremely, extremely strong. It's one of the strongest interactions known in biology. And the advantage of this system is not only that binding interacts very tightly, that it's a tetramer. I mean, Avidin composed of 4 identical subunits, each one containing the active site for binding 1, 2, 3, 4. It's clear that using this system, you can cluster 4 strands or 4 molecules. So this is the main, so again, what are the stages of the synthesis? As I said, we can synthesize long double-stranded molecules from short template primer system. We can also use labeled ones. We can order from chemical company this strand, this G strand, for example, in this particular case, which is labeled with biotin or SH group, or any other residue can be added to this or that end. What's happened then, we use the same strategy for elongation or extending these molecules into long polymer, and again, in the synthesized molecule, you have this biotin again. So now, the second stage is clustering. You interact this tetramer by Avidin tetramer with those molecules producing a complex. In the complex, you have Avidin and 4 strands of the synthesized DNA connected to it. Well, third stage is separation of the C strands. You can see here strands which are separate. I'll talk about this process in more details a bit later. And then self-assembling of those 4 G strands, long ones connected to Avidin, into quadruplex or 4-stranded G4 molecule. Now, a bit in details. As I said, we used this template primer system. The difference between the first moving slide is that we have biotin connected covalently to this end of the G strand. Again, polymerase does a job in the presence of GDP and CV. It's extending molecules to up to whatever. And so, but in this molecule, we have biotin connected covalently to G strand of this duplex. Now, what's coming next? We mix this labeled or functionalized with biotin molecules with Avidin in the ratio of 1 to 4. The ratio can be more. We need to saturate those binding sites. And what's happened? Biotin interacts with those sites, and you produce a complex where 4 double-stranded poly-G poly-C molecules clustered are interacting with this Avidin system. Now we can see proof coming from AFM. It's just a proposal. You take those molecules, depose them on the surface, and you can see here a bright spot, which is corresponding to Avidin, which is bright, and 4 arms connected to it. So these are the DNA molecules. These are all those stars. So we got to prove that the complex we produced is indeed the complex of 4 DNA molecules and 1 Avidin. Now, it's another stage which is, well, more biochemical, no AFM here. We take this long, with this complex, which you saw just in this previous slide, add alkali, just add some base into the solution. At high pH, the strands, DNA strands are getting apart, they're getting diverse. So they're not, this is not the double-stranded molecules anymore. You can separate, you have C strands, and you have complex of 4 G strands connected to Avidin. So you separate them. The separation is based on a length or on mass. So it's size exclusion. The size of this complex is much larger compared to the size of the single strand, and it's getting out of the column faster compared to this. This is an HPLC size exclusion HPLC. We were able to completely separate this complex from C strands, and what we do then, we just reduce pH. We bring the solution back to pH back to normal, to pH 7, and these 4 strands fold. So again, we started from this complex where 4 double-stranded DNA molecules are attached to Avidin. Again, bright spot and 4 arms. We did all those, those biochemical procedures, including HPLC separation, high pH, and reduced pH. And what we got then? We got a match-shaped structures, where in the match, this head is an Avidin molecule, and this part is, which they're more just is the G4 part, and this is the G4 molecule, which is which we got after self-assembling of 4 G strands connected to Avidin. So here you see the example. You got to prove. You can look at single molecules. You don't need to guess. You just look. Now this is awesome. It's terrific in this kind of instrument, because when students were coming into my lab, and myself, when I was brought into the field, I was approaching the skin and saying, "Wow, what's going on? We see the molecules. It's unbelievable." And this is exactly what's happened. So I'm saying that despite that, it's fun working with FM. You are getting valuable information on the processes and on the structures. So now, getting back to initial motivation, we want our DNA molecule to bring them we want to try to push them to conduct electrical current. And it's a very challenging task. Why? There are many problems on the way. One of those is that in order to measure conductivity, one should place the molecule. This is a DNA molecule, the schematic presentation, of course, in between electrodes, which you can see here. And when you drop this molecule, the problem is that the conductive part, if at all, is located here in the core. I mean, here you have insulating material, which has sugars and phosphates. And even if you succeeded to drop this molecule on the electrode surface, there is a problem for electrons to come from the electrode to the core through this insulating layer. In order to surmount the problem, we decided to use gold and silver nanoparticles, which you can see here. These are ball-shaped structures made of metal atoms, gold or silver. So in this case, in the schematic presentation, gold and silver particles are metals. There is no problems for electrons to go from here to this particle, and from the particle to the DNA, which is connected directly to the particle, all the way through this DNA bridge down to the electrode through the particle. So this was an idea how we can overcome the problem of insulating layer of DNA. So, and we decided to synthesize DNA nanoparticle conjugates. And the next part of the talk will be devoted to synthesis of DNA nanoparticle conjugates. So in this one, we use the same strategy. We started synthesis from short template primer system, which in this case is the C strand or G strand, whatever, doesn't matter in this case, is labeled with SH group, sulfur atoms. Why sulfur? Because sulfur interacts very specifically, tightly with either gold or silver surfaces or metal surface, whatever. So we started this synthesis again using the same experimental strategy. We mix this template primer system with polymerase and GDP and CDP, and polymerase does its job. It extends this short template primer system into very long double-stranded molecule, which in this case contains SH group at one end. So then we mix gold particles with these synthesized molecules, and sulfur interacts with gold, producing this kind of conjugates. Now, what we do next, we just separate. Of course, you produce various complexes. There are some particles which didn't react with DNA. There are monomers where you have one particle per DNA. You have dimers which contain 2 DNA per particle, trimers, tetra, and even pentamers. So, and you mix it with 1-to-1 ratio. And the particle is about 20 nanometers in diameter. I didn't say that. So you run the sample on electrophoresis, and then you cut each band, and you get those molecules out of the gel. There are a number of procedures one can get. I want to emphasize here that even you can produce 6 complex of with 6 and 7. They're minor, but you can do that. You can see separate bands here. Well, this is in the 3, 4, 5, and even 6 right here. So then you should get an evidence that it's not just like a virtual, it's a kind of imagination. They're really dimers, trimers, tetramers, whatever. So what you do, you just electrolyte or get those molecules out of these cutted bands, and you deposit them on the surface and you image. And indeed you see that this particular complex is a one-to-one. You don't see it right here because we didn't image, but here you can see this is a dimer where a particle which you see in a bright spot is connected to 2 DNA arms. Here is a trimer where each particle is connected to 3 arms. Here is a tetramer where 4 arms are connected to the central gold particle. And even here you can see beautiful images of 5 arms connected to pentamers. This is the gold nanoparticles and 5 arms leading to it. So this is clustering of 5 DNA molecules by gold particles. You get a number of those structures. Well, they're funny like sea stars. So again, you got it proved, but you didn't reach the goal. We wanted to get the complex, the conjugate, in which we have 2 particles connected to different ends of the DNA molecule in order further to bring it to the electrodes and measure the conductivity of these conjugate, or in particular DNA. So now we use again the same strategy, but here we have 2 SH groups which are added to opposite ends because we want to connect particles to opposite ends. And again, polymerase, GDP, CDP, extending the molecule and mixing them. In this case, we used silver particles. Well, we can use gold as well, but experiments were made with silver, at least those I'm going to present. So we mixed it with silver. No, actually no difference, but I will show you experiments with silver nanoparticles which are yellow. Gold nanoparticles are red. So now, you mix them again with excess of silver particle, and they interact with SH groups covalently, tightly. So you produce here, again, you run the sample on electrophoresis, and what you see. This is the band, corresponds to initial particles. Here you can see the formation of a new band. Again, you are extracting, or you're just getting this, the material out of this band, of this area of the gel corresponding to the band. You deposit on the surface, and you see dimers. Here's the dimers. Even if you approach this dimer, you blow up the image, you see that not only the particles, which are 20 nanometers in diameter, but also a DNA bridge connecting those particles. So we got these conjugates where 2 particles interact with the same DNA molecule. So we got this. So I'm not going to talk about imaging, about measuring of their conductivity. The experiments are underway in the lab of Dani Parat and some other labs. So we hope that we will finally surmount this problem, finally will get it conducted. And if yes, it will be really great stuff, because then those molecules, we can assemble them, because the ability of DNA to be assembled is enormous, and we will be able to finally get to some kind of nano-electrical device which are based on DNA. Now, well, in addition, I want to show you some other examples. For example, of the formation of nanoparticles inside DNA. Actually, with this experiment, we synthesized the poly-G poly-C molecule the same way as I showed you in the second or third slide at the very beginning, and we loaded it with silver ions. Silver ions has very high affinity to the bases, so they are sitting in the core of the molecule, and these are ions plus. So when you add a source of electrons, which is more hydrate, for example, it delivers electrons to silver ions, and silver ion becomes metal. So what's happened, you see the formation of nanoparticles inside DNA, and you can easily see that it's indeed this process occurs when you just depose the sample again and measure it by AFM. You can see these bright spots are small particles, different size, but this is just actually DNA is a template here. It's templating silver particle which has been formed by reduction of silver atoms by borohydrate. Now, I'm coming to almost, I think, the last slides, and I want to show you that not only DNA can be measured. Here the example, we use bacteriophages, or bacteriophage M30. It's filamentous bacteriophage. You can see it has a shape of a tubing, and here in this tubing you have well, some proteins. You can see them here, which recognize the bacterial membrane or bacterial proteins on bacterial membrane. What bacteriophage does, it infects bacteria, like virus. It's a kind of virus, but viruses are infecting us. These bacteriophages are infecting bacteria, so they're not killing, they're just making them sick. So they do so by means of interaction of these recognition proteins with their membranes and injecting the DNA. DNA is here. You can see in the very core of the phage, I mean, you can see a long DNA molecule, and they're injecting this molecule inside the bacteria, and it starts to produce viruses, and then getting sick. It doesn't want to move, so it's almost died. You know, when you're getting virus, you also, it's feeling, I don't know, well, you can imagine how bacteria feels. So that's the way it goes. Now, if you just prepare these phages and you depose them on the surface, on the microsurface, you will see a mess. You will almost, there is no way you will find a single filamentous phage on the surface, because it's, I mean, it's dirty from the point of view. There are a lot of proteins, bacteria, so it's a really mess. So we invented a couple of purification steps, which includes HPLC, high pressure liquid chromatography, and we succeeded in complete purification of those phages out of this whole mass of dirt. And then when we deposit it on the surface, you see that they're beautiful. I mean, I like this picture. I mean, those phages are beautiful, and they're very uniform, might be except for those which was broken. And you see here also this DNA which was accidentally injected by the phage. I mean, the DNA is single-stranded, but it's supercoiled. That's why you can see it on it. Of course, not as bright as the phage, because the phage is about 6 nanometers in diameter, and these DNA, well, it's about, let's say, 2, even less, because of the interaction with the cantilever of the AFM tip. So those are very bright. I mean, and they're very uniform, so one can even calibrate his AFM instrument if you calculate the length, the total length of the molecule. They're very, very uniform. So, well, I think that's, I'm almost running out of time, and I think I hope that I succeeded. Well, I succeeded to show you that that FM has a relevance to biology, and I hope that biologists will be interested in FM imaging of their samples. Again, I mean, I showed you a very small number of samples and examples of the use of AFM in bioresearch. Of course, we're also measuring complexes of DNA with proteins. You can figure out what is the exact position of the protein binding, where is the binding made, how protein recognizes DNA. You can examine the complex of protein and DNA. So there are a lot of tests. Now, I want to tell you that at the time I was brought to this field, we had some leftover of money coming from European sources. I was coordinating the project, and we were saying, why not we buy AFM? And I was a bit skeptical. I said, well, we will buy an instrument, and we'll not use it. It will just be a piece of data equipment. Well, we use AFM almost every day. And I would say that as you start driving, you think of what car should you buy. You say, okay, I'm going for a very not expensive car, or I'm going to a better car. I would say if you start driving, it's good to have a good experience, and you buy a really good car that you will enjoy driving a lot. And we're enjoying the instrument of NT-MDT very much. I would say truly, 7 years we use this instrument with not even a single need to fix it. It works like a good Swiss watch. And I think this part I would like to, to thank you, the attendees, for joining this webinar, which again I think is a great thing. And thank you for listening. And for those of you who will be interested in, in some specific issues of the synthesis of this research, or will have some ideas to collaborate on certain issues you have, I would be happy to get your mails. And this is my email, university email. And then I think I will now, I'll try to answer your questions if I will succeed to open this question box, if I will have any. So I think just I'm open for questions. I don't see any question. Show me.